Okay, today we're going to demonstrate how to do the Graham stain. The Graham stain was devised back in 1844 by a gentleman by the name of Hans Christian Graham. And this is one of the most important staining methods in microbiology. In the identification process, the Graham stain is usually where we want to start. Now I'm going to employ the primary dye, which is called crystal violet, and we apply that to the organism that's fixed onto the slide. Now the crystal violet dye is allowed to react with the organism for 20 seconds, and after 20 seconds we're going to rinse the primary dye, or the crystal violet, off.
What does the crystal violet do?
The crystal violet stains all organisms. What we refer to as gram-positive and gram-negative both stain with the crystal violet. So after applying the crystal violet, we're going to rinse it off with water. Now we're going to add the mordant, which is iodine. And it also goes on for about 20 seconds. The purpose of the iodine, or the mordant, is to strengthen the complex that's formed between the cell wall and the crystal violet. So after the appropriate time period, we're going to rinse off the iodine. The third step is to use the decolorizer. And we use acetone alcohol as our decolorizer. This is the critical step in performing the Graham stain. The decolorizer is going to remove the crystal violet from gram-negative organisms, whereas gram-positive organisms retain the crystal violet. And whether the crystal violet is retained or lost is dependent upon the type of cell wall, the outer covering of the bacterium. So the decolorizer goes on for 2 to 5 seconds, and then it's rinsed off. Now we have the secondary dye. The secondary dye is safranin, sodium safranin. And the gram-negative organisms, which have been decolorized with the acetone alcohol solution, are now going to be colored with the safranin. Now the safranin has a red color. So it's going to impart a red color to the gram-negative organisms. And remember that we said that the gram-positive organisms will retain the crystal violet. They'll still be purple. They don't lose that purple color. So after 10 seconds, it's only 10 seconds for the safranin, then we can rinse the slide, rinse off the secondary dye. And now our slides are ready to be read under the microscope. I like to blot the slide with a paper towel because we have to allow the slides to dry, and blotting off the excess water simply allows the slides to be read quicker than if we let it dry. Simply let the slide dry with just the air. So I like to blot it a couple times to make sure my slide stays dry. And now at this point, our slide is ready to be read using a microscope. And that is the Gram stain method for staining bacteria.